want to say a very big thank you for making it available. I just got home now and I have to commence the class. So today we shall be looking at the long-awaited bottom part of the training, which is molecular docking. So far, God has helped us. We have looked at structural design and modeling. We have learned how to draw structures, design, how to model them. We have also learned how to optimize them, how to save them. We have successfully learned how to model and optimize nanoparticle. If your interest in research is in nano design, nanotechnology, we also went further to look at chemoinformatics. We dealt with chemoinformatics, and now we are looking at molecular docking. In this molecular docking, we shall be covering two courses. Two courses. In fact, so many courses, so many sub courses will be treated under molecular docking. So this part will run from now till October ending. So it will run for more than 1 month because the class is supposed to terminate by October, towards the ending of October. So I want to appreciate us for pulling strong till this very moment. We have really stayed strong. So we begin by what is molecular docking. We will begin by looking at the concept of molecular docking. If we actually talk about molecular docking, because everywhere you go, you hear molecular docking is everywhere. What is really the concept? What is the ideology? So molecular docking it simply means, okay, let me just start with the word docking. Docking simply means an interaction. So once you hear docking means interaction, the study of interaction between two things. When two things are interacting, we say they are docking. That is that word docking. It means an interaction between two things. But you then ask yourself, what are those two things? Those two things that are interacting. If these two things are molecule, if the two things that are interacting is if they are molecule, so we now call it molecular docking. So that molecular docking now means an interaction between two molecules. So the idea behind docking is that there must be an interaction. So anytime you think of molecular docking, maybe anytime you want to apply docking in any research, you must always ask yourself, does this research involve interaction? What are the two things that are interacting? Last week I started a training for one of the big names in my school there, Nekede. She happens to be the director of research and development, so she employed my service to train her on computational chemistry and biology. So but her field is in agriculture. So then she was now asking, how can we apply docking in agriculture? So this was the idea I shared with her. Anytime you are having a research work that has to do with interaction, so I was telling her, now begin to think of that area in agriculture that has to do with interaction. So she mentioned climate change. how does climate change affects the growth of plants. You can see if such work can be done. But at the end of the day, we came up with a paper, and that paper will be presented by next month in Ghana. That paper was, we looked at how we can mitigate climate change. So in that work, we used carbon dioxide. We looked for some microorganisms that can capture carbon dioxide from the air. You know, the presence of carbon dioxide in air causes global warming, because carbon dioxide retains heat. So that is why excessive release of carbon dioxide in air will cause heat, global warming. So that is why we are now saying that one of the ways of reducing global warming is by planting of trees, because these trees now will help absorb the absorb the carbon dioxide from air trapped to the plant through their stomata, the leaves, and uses for their photosynthesis. So we now develop okay, can we get some engineered microorganisms that can actually help in trapping carbon dioxide. So you can see such a work. This is from the idea of nitrobacter, because nitrobatters trap nitrogen and feast it into the root of the plant. The same way too, we can come up with carbon batters. No, this ideology has not been in existence. There's nothing like carbon batters, like organisms that trap carbon, but we have nitrobatters. So we came up with that work last week, and we finished it. It will be published soon. In that work, we saw that some microorganisms like bacillus, some of the species of bacillus can help trap carbon dioxide, because there was an interaction between the carbon dioxide and the organism. So once whatever you are studying has to do with interaction, then doctrine can be applied. So the doctrine is not only limited to drug design, it's applied in all areas of science. I've used it in biotechnology, I've used it in bioethanol production, I've used it in biodiesel production, whereby we use some microorganisms to speed up the conversion process of volatile fatty acid or volatile organic compound, VOC, degrading them into esters. And once they have been degraded to esters, esters are the chief ingredient in your biodiesel. All diesels are mostly of esters. So we apply that ideology of molecular docking in bioethanol production, in biodiesel. So many work outside drug design, because some of us we just think that this is a medical something. They are not only applied in medicine, they're not only applied in drug design. Although the ideology initially, when this thing came up, the ideology when this thing came up, it was on drug design. But when I begin to on my own understand, I've done a lot of work on computational chemistry and biology. I've done several work. So when I begin to look at the understanding, when I begin to understand docking, I can now apply it in several areas. I've applied it in biodegradation of heavy metals, where we studied the ability of some microorganisms to degrade heavy metals. Not just heavy metals, have applied in biodegradation of environmental pollutants. Recently, I did a work with some lecturers in the Imo State University of Agriculture and environment. In that work, we are trying to remediate these polycyclic hydrocarbons from crude oil soils. I've also done the work on PCB, polychlorinated biphenyls. These are organic pollutants. Using docking. Recently, there is a work we are trying to design. I and Dr. Mrs. Chinyere Okafor in chemistry department. In that work, what we are doing was trying to degrade hydrocarbons using docking. We are trying to degrade hydrocarbons from crude oil polluted soils. trying to see how some microorganism can degrade them. Biodegradation, bioremediation of crude oil, hydrocarbon. So, and the work is on progress. So it is, once there is an interaction, once you think of an interaction between two things, then bring in doctrine. Doctrine can be applied there. But once there is no interaction, then forget about it. Doctrine cannot be used. So it has vast application. So it is an interaction, but since it's an interaction between two molecules, what kind of molecules are involved? These molecules will not just be ordinary molecules. It must involve a macromolecule, which is a small molecule, a micromolecule, and a macromolecule. So the interaction is usually between a micromolecule and a macromolecule. How a small molecule, which we call ligand, how it will interact with a macromolecule, we call it protein or receptor. Receptor is more in disease, you can call it protein. So the interaction between a ligand and a protein is what we call molecular doctrine, because these two things are molecules. The ligand is a molecule, the protein is a molecule. So when these molecules are interacting together, we call it molecular doctrine. So how do we now make use of it? From this interaction, they will bind, because the interaction involves binding. So as they bind, as they are interacting, they will cleave to each other. They will cleave. So the degree, let me use the word, the degree of the binding, or the affinity of the binding, how close were they binded, how effective were they fitted, is actually what we are interested in. Because the more you can bind, then the more effective you are on that scope, on that purpose of research. So let's say we want to come up with a drug for cancer, and then we are using phytochemicals, or in fact there's this work I'm designing, it's a PhD work. We want to come up with a new antibiotics. So we are extracting secondary metabolites from some microorganisms. Now we are no more using phytochemical, we are extracting secondary metabolites from microorganisms. When we extract it, we will now see the ability of these secondary metabolites to inhibit the growth of other microorganisms. The idea came from penicillin, where penicillin is a metabolite secreted by fungi, the fungi penicillium, and this penicillin now can inhibit the growth of other microorganisms. So if we can get these metabolites of all phytochemicals and want to study them against cancer, we can now look at cancer cell lines and the genes expressed, or the proteins of this cancer. So if we can do a docking, an interaction between our plant chemicals to the receptor target of this maybe breast cancer. Now, since our idea is on the binding affinity, the bioactive compound that we bind more to this receptor target shows that one is more effective than the others. So in this docking now, you can screen large compounds. My first work, I did 1,000 compounds. You can screen large compounds. That's why we call it virtual screening. Before, we are using HTS, which is high throughput screening. But now HTS has faded. What we're now doing is virtual screening, where we screen large libraries, a library of compound. We screen them against a target at once. From there, each of them they will bind. The one that has the highest binding affinity is happens to be more effective than the rest. Happens to be more effective than the rest. So that is the whole ideology of molecular docking, right? So it is a computational tools because you use computers to predict this binding, right? Are you with me? Hello, are you following?
Yeah, I'm here. I'm here.
All right. So um, all right. So I'm coming. So such interaction now is very essential to us. It's very essential to us in the diverse field of study. So we'll proceed to the goal.
Is the class still on?
Yes, can you hear me? You can hear me, right?
Yeah, I can hear you.
Uh-huh. So the goal of molecular docking is to identify the orientation. I've said it this to identify the binding affinity of the ligand within the active site, within the target active site. So this interaction does not just occur at anywhere. When we are interacting our ligand to a protein, the protein is a complex structure because usually the quaternary structure. We are told in basic biochemistry or in basic food chemistry that when it comes to proteins are made up of monomeric unit of amino acids. When these amino acids, two of them combines, it forms a peptide, and they're held by a peptide bond. Then they form some structures, which we have four structures. These structures are primary structure, secondary structure, tertiary structure, and the quaternary structure of protein. So now they are complex. So since they are complex, some of them are they are made up of a large number of amino acids. Some of them are made up of 200 amino acids. Some of them are made up of 400 amino acids. This interaction now will not just occur at all parts of the protein. It will occur at a given part. It will occur at a given part that will give a better binding. So that part which the interaction will occur is called the active site of that protein. So a protein usually have active sites. So as time goes on, we will learn how to determine the active sites of a protein. Because if you conduct a binding with the parts that are not actively involved in the interaction, then you expect a lower binding affinity, lower result. That is a poor result. So one basic thing is being able to identify the active site of that your target. So the whole goal is at the end, let's get the binding affinity, the binding affinity of our ligand within the active site of our protein. Now this whole process simulates molecular recognition in silico and provides a foundation for rational design of therapeutic and aerochemical. So all these ones are not really important before us. Like I said, I have already outlined different areas of application. We already know its application in drug design, but outside drug design, I've mentioned some of my research work which I've done with docking, so can be applied in diverse areas. Not only limited to drug design. It's applied, so can be applied in diverse areas. No, press the phone. I know the part. So let's look at okay. For instance, docking has been used in the development of SARS. Now, one of the importance of this is that during that time of COVID, they used docking. They use docking to screen new drugs. They were testing. I remember there was a time we heard about chloroquine. That chloroquine is effective for controlling the COVID, the SARS-CoV. At that time, nobody were doing conventional work, professional work. Everybody were using in silico tools. They use docking to discover new antiviral drugs. So you see that it helps you screen a lot of compounds against a given target. So the docking is really important. Similarly, docking has been used to identify natural flavonoids. These are some of the instances where we have applied molecular docking. Now, we quickly look at the types of docking. There are basically three types of docking. There are three types of docking. We have the rigid docking. We have rigid docking. We have flexible docking, and we have the semi-flexible docking. So we have the rigid docking, the flexible docking, and the semi-flexible docking. So these are the three types of docking. But very common to us is the rigid docking. Now, what does rigid docking involve? When you look at the word rigid, it means that both the protein and the ligand are rigid. Rigid means that they are fixed. They have fixed position. The ligand does not move. The ligand does not change rotation. The protein also does not rotate. So it's just like we have a particular site of ligands that bind to a particular site of the protein. So both the ligand and the protein are fixed. We can develop a drug that only the OH group of my ligand will bind to a given binding site of the protein. So such interaction whereby only the OH group.
And what these people normally do is that this company that buys the paddy one, once it's raining, they take it as an advantage to bring the price down, because those people processing it locally will be buying.
Are we following?
Yeah.
Network left you. We are here.
Okay, network left me. I didn't even know. To which point?
Network disappear for your side.
You made mention of rigid, flexible. The last one.
Okay.
You said rigid.
Okay, but are you seeing my screen now?
No. Okay.
I can see you.
Okay, so I mentioned
the types.
I was mentioning the types of docking, which is a rigid, flexible, and semi-flexible docking. So these are the three types of docking. Now I was explaining about the rigid. That for the rigid docking, both the protein and the ligand are kept constant. There is no change, no sterimetric change. What I mean is that we have a defined binding site from the protein and a defined binding site from the ligand. We can say, okay, that this is our ligand, that the amino group of our ligand will bind to a given particular binding site of the protein. So even if my ligand has both amino group and carboxylic acid group, we can define the binding that only the amino group will bind to that given particular particular binding site of the ligand. So we don't expect any other part of the ligand to take part in the docking. We just want the ligand to be rigid, and we want the protein also to be rigid, that the ligand will not also bind to other parts of the protein except that defined part. Now in that case, it is called a rigid docking. It is called a rigid docking. So, now when you are doing docking with rigid docking, it becomes very fast. It does not waste time because the binding site of the ligand is defined, the binding site of the protein is defined. So there is no rotation, there is no conformation, they just go straight and bind. But it has limitation because that particular site of the ligand that will be defined to bind to a particular site of the protein might not be effective in the binding. It may not be effective in the binding, but because of that, it's not possible to change conformation. So that's what we actually mean by rigid binding. But if we assume that it is not rigid, if it happens not to be rigid, that means that ligand can change conformation, but yet at the same active site. So when the ligand is able to change conformation and also the protein does not have a defined target site, it means that any part of the ligand can bind to any part of the protein. Such kind of docking now is what we refer to as flexible docking. Yes, that's what we now mean by flexible docking. Here the ligand is flexible and is rotatable. The bonds are rotatable. It can turn, it can change conformation, and this makes it more realistic than rigid docking. So for example, we can use flexible docking to discover new to identify novel inhibitors. This is a work that I've done before, whereby we work on Alzheimer's disease. This work was done March. I did a work on Alzheimer's disease. That was on March with anatomy department under through the HOD Dr. Stanley. So through their Dr. Stanley Elomore, he invited my team. We came to anatomy department. So we did a work on Alzheimer's disease. They did the anatomy part of that work. They conducted histology on the pancreas and the liver with other parts, with other parts. But we now use flexible docking. We use a flexible docking to see how some of our compound, we used Oda. Yes, that was a plant we used, Oda. This Xylopia, yes, Xylopia authentica, something of that nature. We use Oda, trying to see if Oda can affect the brain, if it can cause or treat Alzheimer's disease. So now such a docking whereby the ligand can rotate but the protein is still fixed is called flexible docking. Now most time the protein also may not be fixed in as much as, but then why we always keep our protein fixed, like I said earlier, that not all parts of the protein are involved in the docking. So before we dock, we identify that part of the protein that will give us, that is the binding site. Every disease has a binding site based on the lock and key principle. So when we identify that binding site of the protein, we make it fixed, but the ligand can now rotate. So we can call it the flexible docking. Now we have semi-flexible docking. Now both the ligand and the side chains of the amino acid within the protein are flexible. So here now, both our protein and the ligand is now flexible. That is semi-flexible docking. Let us not get confused. For rigid docking, the ligand and protein are rigid. For flexible docking, the protein is rigid, but the ligand is flexible. It can rotate. But whenever our ligand can rotate and our protein can also change positions, can change active site, is now called semi-flexible docking. So this part is rare. The part we always use is flexible docking. That is the part, that is the one that is most common. Now we don't decide it. We are not the one to decide what kind of docking we shall be using, but it's being determined by the kind of docking software you are using. So those people that built the software, they were the one that built it and designed it in such way, in such manner, if it will operate by rigid, or if it will operate by flexible. So it is not you that will actually choose the kind of docking procedure that that will be employed. But most of, not even most, all of these softwares for docking, they operate on flexible docking, because we define the active site dimension, we define the active site of the protein. Then the ligand can now have different conformations. That is why if you, by the time we proceed, we'll be having multiple binding results for one ligand, because as a ligand binds, it will bind differently at that same active site, but with different conformations. So the one that will give the best results, the highest results, is what we pick. So we shall come to that extent. I've talked about the semi-flexible docking, whereby both the ligand and the amino acids are kept flexible. So this method balances accuracy and computational cost. We would have said that this method is actually okay, but then because we have kept the ligand flexible already, and we have kept the protein flexible, it will take time. The docking process always take time, and this would have been the best. In fact, this is the best. Why this is the best? Because we defined the active site of the protein by some models, or by the presence of a cold crystallized ligand. Because what will guide us on determining the active site of a protein is what we call cold crystal ligand, ligand that we are used to model the protein. So that active site that the people that modeled the protein, the active site they used in studying the protein, is usually the active site we also use in our docking. But they may not actually be perfect, they may not actually be right. There might be other active sites that are better than the one proposed. So we then see how we can use semi-flexible docking, but the softwares available, or they always operate on the flexible docking. But I've used the semi-flexible docking once. There was a work I presented at PPSN Southeast chapter, like the whole Southeast region. That was around March, something, yes, around that April May, yes, around April May at IMSU. In that work, I used docking to come up with a new insecticide. I used garlic. I selected the organosulfur compounds in garlic, because these organosulfur compounds, they have high insecticidal activity. So I used about 10 organosulfur compounds from garlic. I screened them against 24 olfactory ligands, against 24 proteins that are responsible for this olfactory and lungs in insects, mostly on mosquito. So I did that work, but the software I used was MOE. That MOE operated on semi-flexible docking, and the result was actually okay. Yes, it was okay because our idea is to come up with a new insecticide that we can spray as perfume. We can spray it and still remain there, and yet it will not affect us, but it will affect the insect, because most of the insecticide available in the market, they will tell you when you spray it, you go out for like 30 minutes or 1 hour before you come
It
back.
does so.
Yes, and most of this insecticide, they affect our own lungs. They affect our lungs. Instead of this insecticide to kill the mosquito, they will start killing us. So that was
Uh,
that was a challenge, and one of my co-authors suffered from such a disease as a result of the staying close to where they spray the insecticide. So that moved
you
me
see,
to do that work. And at the end of the work, we got a novel compound, ajoin. Ajoin was a very novel compound found in garlic, and ajoin was able to inhibit these olfactory proteins found in insects. So that work is still in
but the work,
eh?
sorry, the work,
Yes,
can't you put patent it and then give it
yes,
to company to produce?
yes. It's on progress. It's on progress. That particular work is on progress. The only issues we had is that that ajoin had skin sensitization is too high. The skin
Okay.
toxicity is very high, so we are modifying the structure. We are optimizing the structure, and then it causes eye irritation, of course, which is not good. We
It's
something
not
that
good.
we are spreading should not cause eye irritation, or
Irritation.
it
No,
should not
no,
be
no.
sensitive to the screen. So we are modifying the structure, and what we have really succeeded, because the modified structure is actually okay. So we just need funds. In case you know one big man, I beg, just refer me to one big man where you know. We like screen
Wait,
well well.
wait. This
Yeah,
product you people had, eh?
yeah,
You know, it can be patent and
yes,
then give it to a company to
yes,
produce.
yes. And
You
once
understand?
they use
So
it in their
fine.
production, they pay us in every of their products.
Yes, that's what we are talking about patents,
Yes, yes,
patency.
yes.
You know.
What I want to say is that this modified part of the compound now you're talking about,
Mm.
is it still potent like the initial one that has irritating with the eye and skin?
It was more
Because
better,
I
safe.
believe that
It was far better than it.
when it comes to inhibition of the olfactory
Yes,
protein of the
yes.
insect,
Because after modification, we went back again to screen on these 24 proteins, and the result was
and
better than,
it still bind very well.
yeah, yes, and better than the original natural product. But then the one we are modified is no longer a natural product. It's now a semi-natural product.
Yeah,
Yeah.
because it was
That's very
extracted and
natural.
modified.
And modified, yes, good.
Okay, it's all right. Then I wanted to ask you a question, but then you have not finished. I don't know.
No, you can
I've
ask.
asked you this thing before. I've asked you this thing before.
Uh,
You know, when we talk about these active sites of protein,
yeah,
there are in a protein there are so many sites. There is a site in a protein we call allosteric site.
yeah,
It's not actually the active site, but in some cases it regulates the activity of the active site.
yeah,
There are some analog that will bind to an allosteric site. It will now activate, or should I say, influence the binding activity of the active site.
mm,
And there are some cases it will, decrease or inhibit the binding of the active site. Now, my question is, in a, let's say, in a native protein,
yeah,
one that is not being crystallized with any analog,
yeah.
how do you identify active site.
Okay.
Bearing in mind that that protein could have allosteric site.
Yes.
Bear in mind that protein will do what?
We have allosteric site.
Would have allosteric site.
Okay,
Which
the
is
allosteric
different
site
from
is
the active
the
site.
preventer.
No, it's not a preventer. This thing is like, how will I even put it now? Okay, let me use an instance now. You have in your mouth, you have two openings. The one that leads to the lung, the trachea, I think that's what it's called, and the esophagus.
Esophagus, yeah.
Before any substance go down any of these two pipes, one must be closed.
One must close.
So that is exactly how it happened in protein. Protein could have multiple sites, and one could be the active site, other could be the allosteric site. And that allosteric site governs the binding of the active site. In some case, when substance binds to the allosteric site, it will inhibit the binding of another substance in the active site. And sometimes when this happens in the case of hemoglobin and carbon 4 oxide. When carbon 4 oxide binds with hemoglobin, it prevents the oxygen from binding. So that is why the body system try as much as to remove them, because once the concentration is too high, they will now form what we call competitive binding.
Yeah, that's true.
So once this happens, then my question in all this story is, how do I identify the active site? Having in mind that a protein could have active site and an allosteric site. Yeah. Okay. So that I will not mistake the active site with the allosteric.
Yeah, allosteric site. Yeah. Right. Okay, it's quickly is an interesting question, and there are two answers. I know you are familiar with the first one, and that the first one will not be of your interest. Your interest will be the second one. The first one is using a protein with a native ligand, which we are familiar with. But then, if I have a protein that does not have a native ligand, because not all protein are co-crystals. So if I had...
What do you mean by native ligand?
A ligand that was used to model the protein.
Model a particular protein and then store it in PDB.
If
you are browsing some of these...
When that protein was modeled, the ligand that was used to test that protein, let me use that word, the ligand that was used to test the activity of that protein, after modeling the protein, they use the ligand to see how effective that protein will be. So they had to join that ligand with the protein in PDB format. Now,
to form a complex.
Yes, to form a complex. So that means that if you are using such proteins that has that co-crystal ligand, it means that anywhere that co-crystal ligand binds, that is the best binding site, active site for that particular protein, because it has been tested by those people who modeled the protein. Now, but we advise in professional work, we advise that we work with proteins that are co-crystallized. Now, this is actually the reason now. This problem is the reason. So that, one, we can easily identify the active site, and two, we can validate. Though I will come to that part, we can validate our binding results. But not all protein. Like I was doing a work. I was supervising a work from a lady. She's doing a master's in Sweden. She's in bioinformatics department in one of the schools in Sweden. So she contacted me to assist her in her research work. So she's working on colon cancer, and she's using Camellia sinensis, which is a Chinese tea. So in that work, we are working on 10 proteins, 10 colon targets, which were identified from network pharmacology. But at the end, we saw that 2 out of these 10 proteins does not have cold crystal ligand. We have searched from that gene. We have searched the gene, proteins that has that gene in particular. So we searched all the proteins that has the gene that was identified. We saw that they don't have cold crystal ligand. So how do we identify the binding site? That is purely the question that our guy is now asking, because there is a need. So what did I did in that work? Now what I did is that BioVel Discovery Studio, which is one of the tools we'll be using, can identify binding sites. Now initially I thought it was whining. Yes. Now BioVel Discovery Studio can identify the active site of a protein. I don't know the principle that it operates. Now when I try to validate it, I use Pyres. Pyres is one of my docking tools. I used AutoDock. Now when I used Pyres, and Pyres can automatically determine the binding site for you. So I discovered that the same.
Is it the Pyres we installed?
Yes, the one you installed. Pyres can automatically determine the active binding site. So when I look at the binding site from Pyres and the binding site determined by Discovery Studio, because we can teach how to use Discovery Studio to determine the binding site. Now we saw that their difference are not much. Like in Discovery Studio, I got something like minus, because it's always in XYZ in 3D dimension. I got a value of minus 6.1, minus 6.115. Why in my Pyres I got minus 6.0015 or 0012. So you see that the difference is not much for just one of the axes. So when I discovered this thing, and I was thinking that it was actually false, using Discovery Studio to determine binding site. I thought it was false. Now I had to also use AutoDock, although we will not be exposed to AutoDock because it's time consuming and it has to do with small programming. When I used my AutoDock and my AutoDock Vina, it was still the same closed binding site. So I cannot conclude that. And again, even when I used those proteins that have native ligand, the same binding site that was predicted for those proteins that has native ligand, I now assume that this protein does not have native ligand. So I use my Discovery Studio and I got close range, because definitely you don't have 100%. You don't have accuracy.
The same geometry.
Yes, the same, right? Very close geometry.
Okay.
Yes,
okay.
The precision was high, but you will not expect the same value. 5 here to be 5 here and 5 there. But yes,
I understand. You know, the geometry has to be in XYZ.
Yes,
so
good.
There's a tendency that one of it may not be exactly
good.
Where it, you know, this is a kind of key and lock.
Yes.
So any all these phones that have this lock, anybody that resemble you, so far there is a close resemblance.
Good.
Can open.
Yeah, can open. Yes. So that was. Yes. So we can.
Anybody that resemble you.
Yes. Now, once there is a close resemblance, Yeah, that is why this face lock security is not that top security. The most top one is fingerprint.
Yes, now it's working
Mm.
by face, which is face recognition. So anybody that lose that have
What that thing makes use of it is the position of your
your
eye
eye,
and your
good,
nose,
yeah, your nose,
together
yeah,
with the mouth.
yeah.
Once it mentions the dimension and
Okay.
it rhymes with the one you say, to open it.
It has gone.
Okay, it's okay.
Yeah.
Coco.
It measures the dimension, the distance between your eye to nose, distance between your
Between
nose.
the eye and the nose.
Yes, that is what it measures.
Okay. So we in the next 7 minutes which we have left. So when we get to hands-on training, when we get to protein preparation, then we shall now see how we can use that BioVer Discovery Studio in determining the binding site of a protein. So we have seen the different types. Now there are other types of docking which are not necessary to us. We have the induced fit docking. That is not necessary to us. We have the blind docking. Blind docking, some persons do blind docking as like in our titration, we have the first titer which is our rough. So some persons do blind docking as our rough. In blind docking, we don't choose the active site. We just allow it to dock. After docking, we'll see different active sites. From there, we can now see which of the active sites has the best results. Which of the active site has the best result. That is actually blind docking. This is used when the binding site of a protein is unknown. So because we don't know the binding site of that protein, we can now say, okay, let's do a blind docking. The entire protein surface will be scanned. The entire surfaces will be scanned. The docking will take place at the entire surfaces with different binding, with different binding active site dimension. So we'll now check which of the active sites, which of the different active sites that gave a better binding result. We can now conclude that that part is the active site that will be used. But then you have to validate it by screening large libraries of compound. So by the time you screen large libraries of compound, and that same active site that gave the highest binding result keep giving the highest binding result, then that site becomes the active site for that protein. So I used that work before. I've done blind docking. In that blind docking, I use curcumin from turmeric, from curcuma longa, from turmeric. I use it against different bacterial proteins just to identify the best binding site for my antibacterial studies due to the issue of AMR, the antimicrobial resistance. So that was when I used that pattern. But it's just we can still apply this pattern. It can still solve the question that our boss asked, but then this will stress you. Research is always stressful, so we don't have option than to take up the stress. Research is always stressful.
Sir,
Yeah,
can you hear me?
yeah, I can hear you.
Hello, network.
Yeah, go ahead. I can hear you.
Yeah. That means the work I am doing. Yeah.
Yeah,
The work I am doing. That's my PhD work on the synthesis of synthesis and antimicrobial study of metal complexes of
yeah,
substituted aniline. Yeah.
yeah.
Now I can actually do the docking to know the binding site of this ligand against the protein. So to know whether it can be used as a drug.
Yes, in fact,
Yes, now.
if
Not only that, even the toxicity aspect of it.
if I happen to be
Huh?
your external examiner, if I happen to be a external examiner, I will cancel that work. Sorry, sir. Sorry. I will ask you to go and do docking.
No, because you are. It's not every external examiner know much about docking. No.
Yes.
There are very few.
So I'll ask you to go and do docking. Let us see.
Huh?
I'll ask you to go and do docking. Let us see the extent of the interaction. So I will encourage you, sir. Include docking. Include docking in this your work. I don't know the stage you are now, but
Yes, I had a plan of doing it. That's why I needed this class so that I can be able to do it and publish it as a paper.
So what you do now is you already know how to model nanoparticles. So model those your nanoparticles and keep. Just model them and keep.
Yes, for my metal complex.
Yes.
Yes. It's more or less a nanoparticle. You know why? I have 2 ligands.
Yeah.
Attached to a central metal atom.
Okay. So you can model them. Model them.
I have 2 ligands attaching to a central metal atom.
All right. So you can model them as you have learned. Then when I enter docking, you now do the docking for them and attach it with your result. It will be very superb. That means your work will now be in vitro and in silico. Now, the funny part of docking is that anywhere I go to present docking, nobody asks me question. Nobody asks me question because few of them knows it. But you put me no enter person hand we sabi away.
Nobody asks question. Yes, it's true. Okay, to become in vitro and
in silico.
And what?
And in silico. In silico.
But there is a challenge, because when you meet somebody that doesn't know much about docking, that is when they will now challenge you to go and validate the result in a wet lab to confirm that what you are saying is true. I think that one is more challenging.
Yes, that is why he's already doing wet lab. He's doing in vitro. He's already doing in vitro. So we now include it with in silico. So both of them, anyone the person wants to understand, let him understand. If you don't understand in silico, you understand the in vitro. All right, thank you very much.